Morning, good afternoon, good evening everyone. So we are discussing about unit 1 in B.pharm 6th semester CVS pharmacology. So what we have covered so far, we have covered the electrophysiology of the heart, we have covered the drugs which are used or useful in the treatment of congestive heart failure. We have covered the anti-anginal drugs, the drugs which are used in the management of angina. Then we have in the last lectures we have covered about the drugs which are useful in the as a antiarrhythmic drugs which are used in the treatment of arrhythmia, antiarrhythmic drugs. And also we have covered antihypertensive drugs as well, antihypertensive drugs. These all topics we have covered in the earlier video, electrophysiology of heart, drugs which are used in the treatment of CHF, anti-anginal, antiarrhythmic, antihypertensive drug. Today this is the last topic under cardiovascular pharmacology unit 1, that is the antihyperlipidemic drugs which are used in the management of hyperlipidemia, right. So today we are going to discuss about the drugs which are used in the management of lipids, right. So anti-hyperlipidemic drugs, So there is a lipids are water insoluble, they are combining with the proteins and forming the lipoproteins, right, lipoproteins, and lipoproteins are of various types. There is a HDL we call high density lipoproteins, LDL low density lipoproteins, VLDL very low density lipoproteins. So there are so many lipoproteins are there. HDL, that is the high density lipoproteins, this also we called a good cholesterol, because it's not deposit in the arteries, it's a good cholesterol. If your HDL values is high, if you are going for a test, so it's a good thing. LDL is a bad cholesterol. LDL is your low density lipoproteins, So this is the cholesterol. If it's high, it can deposit in the arteries, it can block the arteries, and patients can be more prone to heart attack, myocardial infarctions or other disease as well. So this is just a basic about lipoproteins, but our major focus in this lecture is about the antihyperlipidemic drugs, the drugs which are useful in the management of these lipids. So one first class of drug is statin, right. So statins are well known drugs which are useful in the management of hyperlipidemia. And what the statin do, what is their mechanism of actions, how they reduce the lipid levels. So statins, particularly there is a HMG-CoA, right, it's convert into mevalonic acids. We are talking about the liver, how cholesterol is biosynthesized, mevalonic acids, and here is a one enzymes which convert HMG-CoA into mevalonic acid is HMG-CoA reductase. This is the enzymes which convert HMG-CoA into mevalonic acids. Mevalonic acids further going to convert into squalene, then it going to convert into lanosterol, and then you will form the cholesterol. The cholesterol is required for various functions, right. It's synthesis of steroids, synthesis of vitamins, it's also required in the cell membrane as well. So this is just a biosynthesis of the cholesterol, and there is a enzymes which helps to convert the HMG-CoA into mevalonic acid. That enzyme is HMG-CoA reductase. So statins are the drugs which block these enzymes. So statins will block these enzymes. So these statins are the competitive inhibitors of HMG-CoA reductase. So when we talk about competitive inhibitors, so that means there is a competitions between two things, right? So minimum two persons are required for the competitions. So here again statins are the competitive inhibitor of HMG-CoA reductase. So what is the other things with which statin has to be compete? HMG-CoA. So there are two things. One is HMG-CoA and another thing is statin. So who won the competitions? Statin won the competitions, and statins will block this HMG-CoA reductase. Otherwise, if it's not blocked, so then this HMG-CoA reductase CoA will convert into mevalonic and ultimately cholesterol will be formed. So in this case, statins will won the competitions because it's a competitive inhibitors of what? Competitive inhibitors of HMG-CoA reductase. So it will block HMG-CoA reductase, and due to that, this cholesterol will not synthesize, right? So when cholesterol is not biosynthesized in the liver, then what will happen? I told you in earlier videos as well, body have a compensatory mechanism. So body will try, right? So how body will try in this case? What the body will do, these hepatocytes, what they will do, hepatocytes are the cells of liver. So what they will do? They will do the transcription translations process. By that process, they will do the synthesis of the receptors, more synthesis of the receptors, and receptors for LDL receptors to take up the cholesterol. So to take up the cholesterol, from where these will take the cholesterol? It will take from your blood circulations. This is your blood where LDL is moving, right? And if LDL concentration is more in your blood circulations, it's going to deposit everywhere, and it will result in various cardiovascular disorders. So it's not good. So now you are giving statins to the patients. So what will happen when you are giving the statin to the patients? So it's going to inhibit the HMG-CoA reductase, and by inhibiting the HMG-CoA reductase, your mevalonic acid will not form, and ultimately your cholesterol will not form. So when your cholesterol will not form, then what will happen? Your hepatocytes, your body will do the compensatory mechanism, and by the compensatory mechanisms, your receptors on the surface of the cells, LDL receptors expression is going to increase, and these increased expression of the receptors will do the absorptions of, or do the uptake of LDL, and LDL is bad cholesterol. So when patients will do the test, their LDL is going to decrease when you are using the statin kind of drugs, right? So this is the mechanism of actions of statin drugs. So if I explain it again, so statin वो दवाइयाँ हैं जो आपकी कोलेस्ट्रॉल को कम करती हैं. पर्टिकुलरली मैंने आपको बताया कोलेस्ट्रॉल मेनली हम दो कैटेगरी में डिवाइड कर रहे हैं, बैड कोलेस्ट्रॉल और गुड कोलेस्ट्रॉल. जो एक कोलेस्ट्रॉल जो कि ख़राब है बॉडी के लिए वो है LDL, लो डेंसिटी लाइपोप्रोटीन. क्यों ख़राब है? क्योंकि ये आपका आर्टरीज में डिपॉजिट हो सकता है और आपकी आर्टरीज को ब्लॉक कर सकता है. तो ब्लॉक होने की वजह से आपको हार्ट अटैक हो सकता है, अदर कार्डियोवैस्कुलर प्रॉब्लम्स हो सकती है, एंजाइना हो सकता है एंड सो ऑन. तो आपने क्या किया, एक दवाई दी जिनका स्टैटिन एंडिंग विद स्टैटिन. तो स्टैटिन क्या करेंगी, HMG-CoA रिडक्टेज एंजाइम को इनहिबिट करेंगी. उसको इनहिबिट करने के बाद कोलेस्ट्रॉल कम हो जाएगा. कोलेस्ट्रॉल कम होगा तो बॉडी कंपनसेटरी मैकेनिज्म के थ्रू क्या करेगी, LDL रिसेप्टर्स को अप रेगुलेट करेगी. LDL के रिसेप्टर्स ज़्यादा होंगे, वो क्या करेंगे, जो ब्लड में LDL आपका घूम रहा है उसको ज़्यादा आपका अपटेक कर लेंगे. अब अगर आपका ब्लड से अपटेक हो गया LDL, तो LDL की कंसंट्रेशन आपकी आर्टरीज में कम हो गई. वही आप चाहते हो कि कम रहे ताकि वो आर्टरीज में कहीं पे डिपॉजिट ना हो, अदरवाइज वो कहीं पे डिपॉजिट हो जाएगा, आर्टरीज को ब्लॉक कर देगा, राइट. So this is your mechanism of actions of statin, how statin works. So under statin there are various drugs. अगर हम ड्रग्स की बात करें तो statins में आपकी there are various statins like simvastatin, rosuvastatin, atorvastatin, there are so many statins, fluvastatin, right. So, drugs which end with the statins, these are the HMG-CO reductase inhibitors, right. But there are a like there is a one hormone somatostatin that is also end with the stin, right. There is a celiastatin, right. There is a tempostatin. So, these are the also the drugs. This is the hormones. Celiastatin is the drugs which we are using in combination with carbapenem. Carbapenem is your antibiotics. Carbapenem is your antibiotics, which is cell wall synthesis inhibitors. It bind with penicillin binding protein and inhibit the cross linking of your peptides. So, carbapenems, we are combining celiastatin with carbapenem. The reason why we are combining these both, this celiastatin is not antibacterial, but carbapenem is antibacterial, but it is going to destroy by your kidney cells. Kidney cells have one enzyme, that is the dihydropeptidase. Dihydropeptidase is a enzyme which is present in the renal systems, in renal, and do the destructions of carbapenem. And to avoid the destructions of the carbapenem by the dihydropeptidase inhibitor, dihydropeptide enzymes, we are adding celiastatin. Celiastatin is a drug which inhibits these enzymes, dihydropeptidase, which is particularly present in the renal. So, by these combinations, your carbapenem is going to be much more effective. So, that is why we are combining the carbapenem with celiastatin. What I am saying is, so in MCQ particularly, they can give you the options following are the HMG-CO reductase inhibitor, except the HMG-CO reductase inhibitors, statin, and they will give the option celiastatin as well. But celiastatin is not a HMG-CO reductase inhibitor. Celiastatin, even though it is a statin, so how you have to remember which are HMG-CO reductase inhibitor? You have to see this VA is there everywhere, which are HMG-CO reductase, whether it is Simba, Rosuva, Atorva, Fluva, there is a VA is there, statin, right. But in this case VA is not there. So, that cannot be a HMG-CO reductase inhibitors have a VA and then statin, right. So, this is about your statin drugs, and adverse effect of the statin is myopathy. They can do the muscle breakdown and rhabdolysis. Rhabdolysis is severe skeletal muscle breakdown. So, myopathy is also associated with the muscles. So, these are the major adverse effect with respect to statin if you are using the statin for a prolonged period of time. So, this is about your one of the drugs, statins, and this is, as I said, celiastatin is a drugs which is, and tempostatin is your anti-cancer drugs. It is also not a HMG-CO reductase inhibitor. If you are getting these statin in the MCQ and question is following are the HMG-CO reductase inhibitors except, then you have to select these options, because even though these are statin, but these are not the HMG-CO reductase inhibitors. Thank you. This is about your statin drugs.